Okay, you ready? Okay, my pleasure to open our series with our first talk. I met Irina the first time last week.
Not true.
No?
You met me 26 years ago. Probably.
26 years
yeah,
ago?
when I was working in psychology department. Actually, I was
Me?
there for...
Was
yes,
I an undergraduate or?
No, I don't know. 26 ago you did something, and I was working for whatever research projects. I was supervising laboratory kind of work for students, etc. So that's how you hired my daughter to babysit your daughter.
I remember that. I remember her daughter.
Yeah, okay, of course, you know.
Very poor autobiographical memory. Irina obviously has an excellent autobiographical memory. So when I look through the topics that Irina has authored on, it sort of resonates throughout the whole department. We have differential psychology and taxonomies, neuroscience, cognitive psychology, clinical psychology, mathematical psychology, evolutional psychology, and complex systems theories. So it covers a large gamut of our department, and I've never read any of her work until last week, and I started reading it, and I think it's going to be a very interesting talk, and I hope everybody will help me in giving her a very warm welcome.
Thank you. So, do you guys hear me? Because I turned my mic on, and I don't know if it works. Does it?
Does it work? Can you hear? Yep.
Yeah, okay, good. Anyway, so let's start from motivational slide, why bother, to what I'm trying to present. So you see that this guy is different from others. And they are nocturnal animals having the same genes. They're supposed to sleep. So, okay, this guy in a sample, and when you have outlier like that, you will try very hard to compare apples to apples. So you actually control for age balance and for sex and for maybe cultural background, etc. So you try to compare apples to apples. However, you still see apples are different. And therefore, if you want to keep this in your sample, you of course will have less statistically significant effects when you process your data if you don't control for this extra diversity. So the idea is to look at these outliers, what they're doing, and perhaps screen them for additional parameters in our biobehavioral studies. And perhaps if you separate them, you actually might receive different functions associated with the parameters that you're studying. So in clinical research and in clinical treatments, right now we are treating clients as black box. Well, in my practice, I do screen them for temperament profiles, but normally people don't. And if they don't, they have to do that in psychotherapy. They have no idea what would work, what doesn't, including medical interventions, psychopharmacology. So basically, psychiatrists nowadays, and I'm married to psychiatrists, I know how it's going, trying one antidepressant, it doesn't work, another antidepressant, it doesn't work. So basically treating clients as experimental rabbits just to see what works, because we need more transparency, basically, right? The idea of having the list of parameters for biobehavioral diversity is old. It's actually, even in professional disciplines of psychology and psychiatry, started quite from the beginning of these disciplines as independent sciences. And great people were working on the lists. Great people were offering the ideas, how we can classify biobehavioral differences. And we have two traditions. Europeans try to derive this list from biomarkers, and that's where I came from Laboratory of Differential Psychophysiology. And North American tradition started doing it by statistical, mostly linear statistics methods to do like factor analysis or structural equation modeling. And we kind of have our criticism of this approach. We don't think Big Five or high top or RDoc have anything more than trivial, but it's okay, it's our take. Anyway, let's focus on biomarkers. So when we focus on biomarkers, we have to see what's going on in our brain when the brain regulates our behavior. Well, brain and the body. If you just take simplified picture of synapse, it's a lot of chemistry going on just to produce the transaction. And mind you, electric transaction, it's that lies that we tell undergraduate students that, brain is operating by electricity. One neuron sends the spikes to another neuron and then another neuron. And then we can have networks of this spiky connected wired and then wired and wired neurons. So basically, by analyzing the lightening up of the structures, brain structures, we can see the biomarkers. Okay, it's only a small percent of the brain activity using electrical transmissions, but 100% of the brain using neurochemistry to regulate our behavior. So neurochemical biomarkers are much more fundamental than electric. And mind you, when you look carefully at this ion ligand synapse that using electric transmission, it's actually done similar to your flashlight. Flashlight is using a battery. Battery is a chemical composite, right? So in order to get electricity, you actually still need chemistry. Same idea is in your electric synapse. And that's basically what's going on inside the cell, but between cells, it's still very contingent neurochemical process when you have a lot of neurotransmitters sitting on the fence, talking to each other and negotiating what we are going to do in terms of transmission or anything other, any kind of regulation. In addition to neurons talking between each other, they have astrocytes. What's interesting about astrocytes? A, they regulate transmission practically of every single neuron. B, they don't produce electricity. They regulate by neurochemical ways. So we kind of need to include them into the picture of behavioral regulation. And it all comes to neurochemistry after that. The first bright insight in terms of ideas that neurochemistry regulates our behavior came, as you know, quite early in civilization, offered by Hippocrates and then developed by Galen. They are medical doctors. They're not philosophers. They're not psychologists. They actually had hands-on medicine. And by the way, they didn't plan to do any psychology. And just by the way, they noticed that there are some biologically based differences based on chemical, what they call imbalances. And this idea that chemistry is fundamental in behavioral regulation was validated by the fact that right now we are using psychopharmacology approaches to adjust behavioral deviations. So there is still some truth into biobehavioral differences based on neurochemistry. And when you look at the groups of modern classifications of mental disorders, such as DSM or ICD, it's almost anecdotally that you see the resemblance to topologies that Hippocrates and Galen suggested, right? Okay, it's more than 4 systems, more than 4 humors. This is a list just of chemical systems that have receptors, and some of them have projections. And of course, we have some neurochemical systems that don't have receptors, but still regulate our behavior. So it's more than 4, and kind of we need taxonomy. We need something that's easy to operate with. How do we handle this mess? Well, in addition to the systems, we have microbiota and cytokines and immune system agents and sunlight affecting our neurochemistry as well. So it's a little bit more items that we want to deal with. And that's what basically is the topic of our research for the past 15 years. We do analytic research, what is known in endocrinology, neurochemistry, and psychopharmacology, what's known about functionality of different neurotransmitters and how we can link it to behavioral patterns in healthy people and psychopathology. So glutamate and GABA, that's the good news. They're all different, right? So glutamate and GABA are kind of working horses in our brain, but they have to be regulated by so-called managers, right? So these managers, they're monoamines and acetylcholine systems. The beauty about it is that they have long projections all over the brain. And this is a unique feature of the systems. So when you look at the list of agents to worry about, you already see that they're functionally different. Some of them work in horses, some of them are managers. Okay, good. So we look at the managers. Problem is, each of them have several functions, at least in the literature. It was linked to several functions. For example, dopamine was linked, some people linked it to good mood, but it's totally linked to motor regulation and to psychosis. Noradrenaline was linked to attention and emotional activation, etc. So it's like several functions on one neurotransmitter. Then each of them have several receptors. Receptors have different locations and different functionality. So in addition to that long list, each of this receptor type should be taken care of, right? Plus, if it's not that bad, they regulate each other. These systems regulate each other. So this kind of contingent soup that we have to entangle to understand what's going on. Okay, so that's a summary slide. David, do you have a printout here handy somewhere?
Rachel has printouts. I think she handed them out.
Yeah, okay. Oh. Guys, yeah, you have the printout, right? If you don't, yeah, great. So basically, that's a take-home message. Literally, physically, you can take this piece of paper home, yeah. It's a summary of what we did, but this slide is very busy. And some of you don't feel like going into all these cockroaches, small things, details, names of the neurotransmitters. So they don't care about, some of you don't care about this composition stuff, right? If you don't, it's fine. I totally understand you. I was hesitating. I'm basically a psychologist by training first. So I was hesitating to go this way. But important part is you need to understand how fundamental these 12 components are. And the rest of the talks, I'm going to talk about how we derived that model, why it's structured this way, not another way, and then give you some examples of applications in research and in clinical practice. Okay? So anyway, let's get first what the main principle of our approach is. The main principle, what we call functional constructivism. So the behavior should be constructed. It's not the push automatic button and you have little rabbit jumping out. No, behavior every single time you have to construct. What do I mean by construction? Well, first we have a lot of degrees of freedom in environment to be attended to. So behaviorism doesn't work. Stimuli reaction, you have stimulations and then you have reactions. Doesn't work. Why? Because you have millions of stimuli which to attend to. Like to do the reaction, you kind of need to sort it out. Plus, body has many degrees of freedom to act, to choose. I mean, I can put the pointer here, and I can approach to it from this side, or I can approach it from that side, and I can toss it, etc. Right? So you have different degrees of freedom to do the same action, and we have to sort it out. So when you're sorting it out, basically you are selecting some alternatives which you like more than others, and suppressing others. So you are suppressing 99% of alternatives. It's a lot of alternatives. That's what your nervous system does. Mostly it's a lot of suppressions of some stuff that you shouldn't do. And then out of these alternatives, you need to sequence them. What goes first, what goes then present it like plan A, this is my favorite plan, plan B, if this is my A, plan A doesn't work, and go on. So you get more details into your planning, and then you need to maintain your choice. And maintenance, that's what we call maintenance, involves a lot of suppression and inhibition. So it's not the same energetic maintenance that we do when we drive a car and keep our leg on the foot on the pedal, on the gas pedal. It's different. It's a suppression of alternatives that we don't like. And if the suppression goes cuckoo and your neurotransmitters can't do it anymore, the behavior becomes disoriented, behavior becomes disorganized, right? So in this case, it's maintenance. That's why serotonin is so important there. Anyway, so we looked at the multidisciplinary kind of studies, and that the main thing is to use multidisciplinary approach. You need to look at different disciplines such as kinesiology, how the behavior is being constructed, functional neurochemistry, functional neurophysiology, of course, psychiatry, clinical studies, etc. So we look at the models of how behavior is being constructed, and all these models have different components. They have different names of components. However, universally, they have a convergence to three aspects of behavior. They are saying we have to differentiate between orientation, integration, which is like selection of alternatives, and that energetic maintenance with a feedback to orientation if needed. So, okay, that's good convergence. When we go to neurochemistry, we find the same convergence in the functionality of monoamine systems and acetylcholine systems. That study of Jacobs was done actually in 1980s, but then, of course, they expanded the studies, etc. But the main approach in Barry Jacobs' lab is to take cats who are freely behaving. They're not in the cages. They can do whatever they want. And implant electrodes in their brain in the neurons that belong to serotonin system or noradrenaline system. They first start from serotonin and then they expand it to noradrenaline. And they monitor what that neurons are doing. Then they expose cats to different pleasures, different negative or strange stimuli. And then they see how behavior is changing and how the blood content is changing to see the sympathetic system response as well. So in all this exposure, cats did what cats are supposed to do. They reacted. They had strong behavioral response. And neurochemically, yes, sympathetic system was changing too. So what the neurons were doing? Noradrenaline neuron tripled or doubled activity, whereas serotonin neuron produced quite repetitive or kind of, rhythmic activity and didn't change much. Well, this is interesting. So they kept introducing different stimuli, thinking, well, serotonin should react somewhere. No, it didn't. It has this maintenance sort of rhythmic activities of neuron, but it didn't change. Interesting part is that when they didn't change the stimuli, but they continued introducing cats to the stimuli, noradrenaline neurons started having less and less firing. And that's how people started understanding that, with, of course, additional research, that noradrenaline neurons release the neurotransmitter in the case of novelty. So novelty makes noradrenaline neurons act and not just any attention, whereas acetylcholine is more for sustained attention. Dopamine is a completely different story. Dopamine is always, not always, but very, very often considered to be a neurotransmitter of happy mood or reward or anything like this. Guys, if you hear that, walk away. This person doesn't know dopamine much, okay? So anyway, dopamine system is interesting because dopamine is getting released in any situation of significance. And in fact, it's more released in negative situations where, of course, you have high significance, such as electric shock or defeat during the games or any kind of other adversity. And theoretically speaking, if you have an increase of dopamine, and if this hypothesis of extraversion is correct, then a person should be more extroverted, right? Or having better mood. It's not what's happening. A person becomes schizophrenic because the salient system is overloaded, right? If you ever talk to schizophrenic people, they assign significance to everything. They assign significance to every stimuli, and that's why paranoia is so common in schizophrenia. And of course, they are not happy. Schizophrenic people are actually depressed and sometimes even suicidal. So let's say that hypothesis of dopamine involvement in extraversion probably should be reconsidered. But if you look at the overall functions of dopamine, it's all about assigning of significance. In cognition, assigning of significance to stimuli, if it's overloaded, you will get psychosis or OCD, for example. In motor behavior, assigning of significance or sequencing of motor actions is noted when that system goes cuckoo in Parkinson's disease, right? So you have motor action dysregulation, tremors, and slowdown. And in motivational processes, release of dopamine helps you to highlight what's going next and what don't go, where don't go, right? So basically, that's why it was linked to that reward behavior, because indeed, dopamine is being released during rewarding behavior, because the person is saying, oh, I want to make it again, I want to take it again. So dopamine is marking that pleasure, of course, to come back to it and maybe repeat it. So yes, partially it's kind of participating in reward, but it's not always. Anyway, Trevor Robbins is famous for studying monoamine systems, and now he, like, for second year in a row, comes in his ratings as #1 in psychology in the world. And he is, by training actually, came from neurochemistry. He was a chair of psychology department in Cambridge University for 16 years, but his first training was in neurochemistry. So when I approached him with our model, he kind of got interested in this and participated. So we finalized this model, and one of these components is this model is his component. He studied functional division of labor between monoamine systems and, of course, acetylcholine systems. And, of course, he's famous, absolutely famous research in OCD and impulsivity. So now we are summarizing that vertical partition of the model. We get the first column orientation more or less one way or another entangled with noradrenaline and adrenaline. Second column is more or less entangled with integration of behavior supervised by dopamine and delta opioid receptors. And serotonin systems and acetylcholine systems are important in sustaining these internal loops that neurotransmitters have to maintain during the chosen alternatives. Now let's go horizontally. See that horizontal line, so we'll go horizontally this way. Horizontal split is actually easy because you can see it neuroanatomically, not only neurochemically. Neuroanatomically, if you look at hypothalamic pituitary system, you can see very nice division between posterior and exterior pituitary and hypothalamic nuclear that regulates physical versus social behavior. And by social, I mean not just, socializing with your peers or using social networks, etc. No, it's a body-to-body regulation. It involves parent-child regulation. It involves prey-predator regulation. So basically monitoring and attuning your behavior to another body. And that's fundamental. That's why you can do it in translational studies on animals if you want to. Like oxytocin, for example, yes, it's a hormone linked to social perception of social support, but it's also released in milking production and in uterus control during delivery, right? So it's all about parent-child sort of interactions as well. And we published a paper analyzing involvement of all these groups because the hormones are playing in teams, right? They're not playing. One hormone doesn't control everything, it's anything. So they're all playing in teams, but they're playing in a very logical way. And that's how we validated the idea that it's possible that empathy and sensation seeking should be separated in terms of temperament. And of course, there is a corresponding symptoms of psychiatric disorders. Now, the top level is here. It's called blue cortical traits. It's practically regulated by that cortical monoamine systems and acetylcholine systems that I just mentioned. But they rely on glutamate and GABA heavily. Without glutamate, we are stupid, right? So glutamate gets you these electrical activities, and these systems shape it, basically, right? So it's not just a single neurotransmitter playing here. So anyway, so now we can see that our behavior is regulated in different sort of cycles of involvement. We have regulators for our behavior. First, in a wider context, that's what cortex for, and that's what monoamines do. And different family of neurotransmitter systems are hormones, which regulates our social, body-to-body cycles of regulators, and physical, it's mostly neuropeptides. So even by families of neurotransmitter systems, we can actually see a nice differentiation that helps us to validate this model. And the final row is that so-called opioid receptor systems that regulate our emotional disposition. Interesting part about opioid receptor systems that they are G protein coupled receptors. Why it's so important? Because most of neurotransmission in the brain doesn't happen, as I say, due to using that ion ligand electric way, but it actually happens through this neurotransmission, through G protein coupled receptors. What they do, they use various mediators as second messengers, and they have this negotiation. And this negotiation can go fast or it can go slow. It can take up to 3 days for the neuron to process, okay, what I'm supposed to do with the signal, right? For 3 days. I mean, train can already kind of leave, but it's still kind of thinking about it. So they're slow, but they're fundamental. All brain works like that, you know? And here, in our case, it's important that exactly opioid receptor systems are controlling our managers. So in a way, it's managers of managers, right? So they are great cardinals regulating our body. And intuitively, you might actually recall that, yeah, when I'm in bad mood, everything in terms of perception and the choice of my alternatives and behavior is affected, right? I'm making decisions based on my bad mood. Or if I'm anxious, everything is affected. And that's basically what opioid receptors can do, because they're regulating our not only decision-making, but the whole chain of construction of behavior, right? So now you can see how fundamental they are. And in addition to that, they are part of our immune system, and they're produced by our gut's microbiota. So here you have echo of Hippocrates talking about, you know, participating of the body in the regulation of behavior. Indeed, and the recent pandemic of COVID did demonstrate that indeed cytokines, which is part of immune system, can regulate your HPA axis, can regulate your anxiety, and can produce chronic anxiety after COVID, right? So it's very important system, opioid receptor system. Now, we have here a graph basically showing that from the construction of behavior, you have a feedback of behavior, saying, okay, yeah, I have to do this, and this is my feedback how I do it, and there is a reasoning here happening on the cortical level, affected by my memory. Okay, I should do this and this, I shouldn't do that and that. That's wishful thinking, right? Wishful thinking. It gets to behavior, and the behavior of alternatives kind of chosen wasn't happy, so it gets back to your consciousness as saying, why did you do that? You shouldn't be doing it. And hormones saying, well, I do it because it's kind of easy. However, during all this process, very often psychologists miss the fact that we have that dispositional influences from our opioid receptor system, from immune system, from gut microbiota, doing direct effect to all these decision makers, right? That's the idea. Anyway, if you look at the brain structures, and if you look how the brain structures are involved, the brain regulates the behavior starting from maintenance. It doesn't start from stimuli, right? Most of the time we are blind and deaf to stimuli. We don't care about stimuli unless we need it. So, but basically we still need to have that maintenance of posture, maintenance of water retention and food retention, etc., and social cycles, etc. So the behavior starts from maintenance. Then when the adjustments are needed, And here, different colors means different levels of regulation. We get this reselection of alternatives, and then if this doesn't work, we have orientation. Okay, so orientation is called on scenery quite rarely, or if you need to continue orienting in novel, variable environments or complex environments. But if environments are not complex or variable, that's it. Your orientation probably is less important. That's the best case scenario. That's a scenario for very flexible behavior, very adjustable behavior, right? But most of behavior is actually automatic, and it's looping on something that we already learned or something that we are prepared. We already choose alternatives, and we just go according to the list to do. That's it. We are not orienting. We're just looping in our choices and just coming back to maintenance. But there is another scenario called impulsivity, when our maintenance systems gave up, they don't maintain anything, and behavior became more disoriented, and behavior is really, really sensitive to orientation, sensitive to distractors, to trigger stimuli from environment. And that's why, people become impulsive and reactive. And this reactivity can be both physical and social, but if you see the top cortical regulation is missing here, because at that point your hormones take over and that's it, and you don't control it. Anyway, that's a summarizing slide, as I said. Now, so what? So now the part is how we can use it in our research practice. Well, the first thing is because most of research in neurochemistry and pharmacology done on animals, it's totally applicable, that model, totally applicable to translational studies. And translational studies means we can translate whatever we receive on animals to humans, right? And that's what we did. David asked me to give a couple of examples. That's what I do. In research with Professor Isai lab in Iran, pregnant rats were addicted to opium. Then they had their babies, and we looked at the maternal behavior. And as you understand, maternal behavior is very complex, right? But if we have that paradigm of what to look for, what criteria to look for, that helps you to sort it out, what behavior belongs to what. And that's what we did. We separated elements of behavior related to endurance and elements of behavior related to ease of integration, such as number of feeding or number of grooming, etc. And of course, we measured neurochemical levels, if there is any changes, and serotonin and BDNF factor, of course, went down predictably, and corticosterone went up. So neurochemically, we knew that the changes are there. And behaviorally, yes, behaviorally, we actually received significant results when maternal endurance and integration actually went down, right? So structuring it in terms of that FET actually helps to analyze your results better and helps even to put the methodology in a little bit more brushed up based on neuroscience way. Now, in healthy humans, right, in healthy humans, we use that test. The test was developed about 35 years ago. Now it's a new version based on the FET structure. Original version had a little bit different structure, and that's you have a printout. The test is completely free for anybody who is doing research. It's translated to 24 languages. It's available online, and it takes literally 2 pages. That's it. That's a test. Really, it's no sweat. Or it takes 10 to 15 minutes on computer. So you just, if you have a person who is willing to complete it, you can send the Excel file to this person. This person is completing the Excel file, send it back to you. You have your data. So really, screening for biobehavioral traits is really possible, but it will give you a good information. Everybody who uses a test, everybody receives some significant results. Everybody, because these scales are chosen based on neuroscience, basically, right? Anyway, these are the scales related to endurance and integration, and these skills are related to orientation and emotional regulation, and that's a history of validation. It was validated over 35 years with different measures. We have more online if you needed to read the whole thing. The beauty about this test is that Excel file. So it's not only compact, it's not only informative, and it's not only free. But I think the best thing out of this test is the fact that it has Excel file, which you can easily use. And once your subject completed the scores, completed the response, right there in the same file, you have a summary sheet that you can print out to the subject and send the subject home happy with some kind of profiling. But if you scroll down, here you have the horizontal line with answers from this person. So you don't need to do anything. You can just, when you're collecting your data into your data sheet, you just copy and paste this data. So A, you don't need to key it in. B, it's already processed, right? So it's very low maintenance if you do the research. But it will give you the idea about temperament profiling. And the benefits of it, I can illustrate using the human subject studies that was done in Cambridge by my colleague Kenan and Trevor Evans. What they did, they used tryptophan depletion. Tryptophan is a precursor of serotonin. So consider it's like low serotonin, right? They artificially made healthy humans having low serotonin for a couple of hours. And these people were engaged in special technique, like a game basically, where a subject was either an agent or, let's say, source of doing something bad, or a victim of that harm. So it's either agent of the harm, responsible for the harm, or a victim of the harm. And what we received when we did the screening for temperament, and the fact is that if you wouldn't do the screening, the statistical effects were really low. But if you did the screening of contrast groups, you actually have some effects that high neuroticism group had sort of loss of shame while being agent. So agents should feel shameful for causing harm. Highly neurotic people under tryptophan depletion lost this shame. Normally they're very neurotic, right? But under that thing, they lost, paradoxically, the shame. But if they were in the victim group, they actually gained the shame. So they felt guilty, even though somebody actually victimized them. And this is very consistent with clinical literature, right? If you, in clinical practice, I'm a clinical practice, right? So we know that universally victims of abuse feel shame. That's why they don't go to us, and that's why it's so hard to get information out of that. They feel shame. So anyway, to explain it, we started playing with our kind of model, and there are four stories here. One story related to what causes high neuroticism, what causes, then second is what causing having guilt or shame, how the neurotransmitters or hormones work. Then feeling of responsibility for harm, and having that serotonin withdrawal, right? So four stories, we need to take care of all of them. And that came with a spectral hypothesis, how this 8 sort of systems, or whatever, 12 systems participate. And it was consistent with our results, right? So basically, we have more transparency of what's happening. It's really pays attention. And Kenan, he didn't play with FET before. He just, what he had to do, basically to add it to his testing package. That's it. And that's what I suggest. You guys add it to the testing package. You will receive your results. We will help you to do the spectral hypothesis. Right? You don't need to learn the neurochemistry from the beginning, you know. And if you use already some sort of tests, which many of you sometimes use, like Big Five, etc., and many people say, look, we already have battery which is saturated by tests. We can't add any other. Okay, at least use that framework in terms of composition. And any test that you use, I can find analogy of the scales that you're using to that FET, because that here is analogies, or let's say consistency between our model and these models related to green traits, basically physical traits, to social traits, to cortical traits, and 2 slides of emotionality. So if you have any test and it has some scales, I can tell you where the analogy to our test is. It's if you just don't want to lose your results and want to understand the results, right? But honestly, I would just use this one. It's much easier. And then the final application is in clinical practice. It's 2 applications, right? One is about taxonomies of psychiatric disorders, and second is about screening for treatment. Well, treatment, I probably don't have much time today, but the taxonomy, okay, let's go back to our FET. Here, a consistent behavioral pattern is CBP, and temperament type is T-CBP, right? So it's about normal behavior. We can have cases going above the norm and below the norm. And this is kind of very rough presentation how you can just, even in this very simple split, you can see consistent patterns that coincide with some mental illnesses. But if you want to go with more details, that's what we did. We just took DSM-4 at that point. Now it's DSM-5, but at that point it was DSM-4, and took every single diagnosis. And I know you don't believe it, but that's what we did. We took every single diagnosis and every symptom of this diagnosis. Symptom of that diagnosis, it's a consistent behavioral pattern. So it's important, right? And we matched it to the components of our FET and suggested, well, it probably should be low here. It's the same here as here. So basically, we had this hypothesis, and if you see references here to the left, it means we actually did the study on this diagnosis. So if you see spectral-wise, you have different spectrum for different diagnoses. And that's the results. And as you see here, for example, a low mental endurance, if you have that component or symptom of inability to focus, it's relevant to mental endurance in our model. Or if you have motor retardation, it's like low motor tempo. If you have expressive language problems, it's your social tempo. So you do have resemblance between FET components and symptoms of psychiatric disorders. And that's the results of our depression study. Then, of course, you have different spectrum for anxiety. We had this generalized anxiety study, kind of confirming that hypothesis. Then, of course, I just going to page down, and you can see visually how different these diagnoses are, right? So here you go, right? So they're just different diagnoses. So you do have different spectrum. So when you put it on one slide, all the spectrum, you can see that there is no 2 identical disorders, but at least you can have the systematic biomarkers-based classification of mental disorders instead of having this brick Bible kind of psychiatry that we have to study on with very repetitive systems, right? Repetitive symptoms. So we think it's kind of efficient this way. And currently we are pleased with the help of our volunteers and students. And that's the list of projects currently that we are working on and trying to prepare analytic reviews related to sex differences in neurochemical systems. And it's funny that every neurochemical system has sex differences. It's interesting. Age differences, we already had student Miranda who worked on teens, and now we have a volunteer working on aging and neurochemical systems. Then topology of aggression, Ms. Gaikalova was quite helpful. We almost done with the paper, but we need to finalize it. Diets, we have students from McMaster University here helping us to look at diets and neurochemical systems. And now we are working on mapping of, in more details, locations of receptors and projections of specific neurochemical systems. Because the truth is that it's funny, I don't know if you know that location of projections doesn't match the location of receptors. You know that many systems have this phenomena. You have projections to certain areas, but you don't have receptors for this neurotransmitter. And that was a puzzle for several decades, and then people suggested maybe it's a volume transmission, maybe it's extracellular transmission that happen to be true. So a lot of transmission happens not through the synapse, not through even electric synapse or biological synapse or chemical synapse, but outside of the cell in extracellular space. Well, I can talk about it forever. But anyway, finally, just last idea that we are toying with to have regional contrasts in terms of factors affecting neurochemical systems. And those factors include, for example, sun exposure to sun, or there is no exposure, immune challenges, or social sort of routines, or sense of loneliness, exposure to drugs, exposure to common psychostimulants. So if you look at the map, actually there are regions that you can contrast. Believe it or not, actually there are regions that have less drugs exposure and more drugs exposure and less common psychostimulants of specific type. So it's possible. So if you do that, you add it up with temperament investigations or even specific investigations for neurotransmitters in a more transparent way, and then you will have a nice transparent map what's happening in terms of neurochemical regulation. Because the truth is that these factors do matter, right? These factors do matter. And we have to keep an eye on this. Anyway, here's my thank you for my colleagues, and summary. Basically, we are after united taxonomy based on neurochemical biomarkers. The models that we developed is applicable in studies on animals, healthy humans, and clinical samples. And we strongly advise you to use our test. And that's 2-3 pages of references. Okay, thank you.
Thank you. And you left a couple of minutes for questions.
Okay, good.
So we have about 5 minutes for questions, if there are any from the audience.
I thought I will never make it. I thought it's exactly 50 minutes.
Yeah.
Wow, actually, maybe I cut something. Oops. Yeah. Okay, guys. Okay, it's very intimidating. I understand it. Okay. So let's agree. I spent on this 15 years. I can bet my money that you didn't spend 15 years. Okay. So if you ask a question, I don't expect you to use the correct terminology or correct name of the neurotransmitters, or even, not asking something trivial. Okay. But please, if you have some kind of warming question coming, yeah, please ask simple stuff. Like, I understand I'm in psychology building, okay? I'm not in neurochemistry. Okay.
Smaller, right?
Yes. Great talk. I was curious how, if we're seeing like some sort of presentation, like a neurochemical presentation, when you have patients with comorbid diagnoses.
Right.
So there seems to be like a pretty, I don't know, distinct presentation for some of like the mood disorders you were describing. But how does that account for, or how is that, I guess, presented in patients with multiple?
Right, comorbid things.
Yeah.
Here you go. We have a comorbid study, comorbid depression and anxiety, and we are totally proud of it, because any other studies conducted with any other tests, such as Big Five or negative positive effect model or anything like this, they didn't differentiate even between depression and anxiety. For all clinical disorders, they have the same pattern with a Big Five or negative positive effect. It's the same pattern. You have high neuroticism and low extraversion. So our model is more picky. You saw it. With comorbid study, we even didn't expect that this model will be efficient. We still did it. So in comorbid study, what we did, we compared that sample. You can see the sample compared depressed people with just generalized anxiety disorder, right? So separate depression group, separate generalized anxiety disorder, and comorbid patients. Because I'm a clinical psychologist, we have our own practice, so we have a clinical sample. And what we did, so of course we used that screening of temperament. We do it for any intake person, so over years, because I'm in the practice like for 20 years, so over years we accumulated quite a lot of profiles. And what we did I find is that it's not just addition, simple addition of whatever symptoms in GID was, like in generalized anxiety, and what symptoms were in depression, but actually it's quite different. It's mostly cortical traits were damaged. Now, if you think about it, how it's possible neurochemically, basically in GID you have dysregulation of kappa opioid receptors that's kind of getting into GID. But I mean, if you want to hear it from me, but okay, I can give it to you. So dysregulation of kappa opioid receptor systems, but not so much effect of mu opioid receptor system. Kappa makes you more aroused, more, let's say, try to search for alternatives. And mu makes you more relaxed and say, come on, approve everything. Don't search, like really, it's hakuna matata, it's everything fine. And all opioid systems actually work on, opioid drugs, let's say, work on that mu opioid receptor system. Anyway, so what's happening, these systems actually suppress each other. So if you have higher kappa, we can expect that your sense of approval will be probably less. But still, if you have quite endorphins, if you have regular exercise, etc., you actually can get more activities, and your generalized anxiety will be not so bad. Now, in depression, you will have endurance and physical tempo going down. Mostly, I suggest serotonin system is actually important. And mu opioid receptor system works with serotonin a lot. So mu goes with serotonin, kappa goes with noradrenaline, actually. So, okay, you have two comps playing together each other. Now you have comorbid. Comorbid is happening not overnight. Most of studies show that comorbid situations start from anxiety, and then the nervous system is getting exhausted, and you don't have enough serotonin to repair everything, and then your depression comes. And that's how you get comorbid, not another way around. It doesn't start from depression, then anxiety appears. So comorbid situation always starts from, I mean, comorbid anxiety disorder and depression disorder, starts from anxiety overwhelming a person. For over a year, the person becomes even more depressed, even when it started just generalized anxiety. But if triggers are not removed and behavior wasn't adjusted, then you have depression. So in this situation, what's happening? It's just your cycles that's supposed to, remember I told you serotonin is needed to suppress alternatives, right? If you are running out of the suppressors, then you will get that sense of that I don't care, because mu opioid receptors are basically stop acting. So the sense of dysphoria, the sense of low motivation. I mean, I can probably give you more details because mu is regulating dopamine, and so you stop planning, you start integrating your behavior.
I'm going to suggest there's a reception up in room 205.
Yeah.
So we can carry on this conversation about comorbidity and the neurochemical underpinnings of.
Okay. Thank you, guys. Thank you for your attention. Thank you so much.